Hello everyone, today we're going to be talking about microbial metabolism. And we're going to be focusing on bacteria. So before we get into the metabolic processes of a bacteria, let us understand the basic structure or the basic components of a bacterial cell. So basic components of a bacterial cell. One, because the bacteria are prokaryotes, they have a nucleoid instead of a nucleus. Secondly, they have plasmids. Moving further down, they have what is called a capsule. A capsule is a jelly-like material that is present in a bacterial cell wall. Then we have pili. Pili are used, their function is movement and to gather food. We also have the cell wall, which is again made out of the peptidoglycan layer. There's an I or L over there, glycan layer. Here we go about the gram-positive and the gram-negative bacteria. If you aren't familiar with the gram-positive and the gram-negative, or you need to brush up, we have a separate video for that. So make sure that you go check that out so that we are all on the same page. So this is like the basic components or the structure of a bacterial cell. Now let's move on to the processes that occur in a bacterial cell. So first of all, let's talk about respiration. Bacteria respire in two different ways. One is the anaerobic, or they respire as fermentation. Fermentation. Okay. As the name suggests, anaerobic means no oxygen. And fermentation, we're going to be focusing on fermentation for now because it is more complicated than anaerobic respiration. So a simple definition of fermentation is taking one sugar molecule, one glucose, and converting it into two pyruvates, and while converting it, producing 2 NADHs and 2 ATP. So this is the basic outline of fermentation. This is how it happens. Needless to say that both of these respirations are not nearly as efficient as the aerobic respiration, because it has oxygen in it, so it makes things a lot easier. Not nearly as efficient as aerobic respiration. So now, while we're talking about fermentation, there are 2 types of fermentation. One type is the alcoholic fermentation. Over here, as the name suggests, the waste product or the end product of this whole cycle, of this whole chemical reaction product, is ethanol. As the name suggests. An example of this kind of fermentation would be yeast. Yeast is used in a lot of fermentation events, even in a kitchen. When your bread rises while baking, it is because of yeast. And yeast is actually a fungi, so it is not a bacteria. The second type of fermentation is called a lactic acid fermentation. And again, as the name suggests, the end product or the waste product is lactic acid. An example of lactic acid fermentation would be our muscle cells. When we work out really hard and our muscles start to ache, it is because even though our muscle cells usually do aerobic respiration, they switch when we start working out really hard. They just can't keep up with the oxygen demand, and so they switch to lactic acid fermentation, and the muscle cells actually form lactic acid, which makes our muscles ache. That is why we feel the pain that we do after a heavy workout. This is also used in by yogurt making bacteria. For the same reasons, when milk thickens, because bacteria perform lactic acid fermentation and it makes yogurt. moving on from that, we're gonna be talking about the nutritional types of bacteria. So, bacterias and many other organisms can be phototrophic, phototrophs. They can be lithotrophic or lithotrophs, and they can be organotrophs. As the name suggests, for each one of them, phototrophs require light to be in order to make food, to get nutrition. Lithotrophs require inorganic materials to get nutrition. Organotrophs, again as the name suggests, they require organic materials to provide for the nutrition demand. Other than that, there are sources of energy that organisms use. And this might look the same, but it is a little different. So one source of energy is the sunlight. Another source of energy is the inorganic compounds. And the other source of energy can be the organic compounds. And while this looks exactly the same, the difference is that these are the nutritional types, and this is the source of energy. So this is where they get their energy from, and this is where they get their nutrition from. Then there are also there is also one other classification, and that is the sources of carbon. So where do they get their carbon from? So it can be from organic compounds, or it can be from carbon fixation. So these are like their in bacteria especially, we can't just say that they are autotrophs or they're heterotrophs, because some bacteria might be phototrophs, but they might be using inorganic compounds for their energy source, and they might be using carbon fixation for their carbon source. So it can change from every species of organism. So talking about their reproduction now, bacteria reproduce through what is known as binary fission. Binary fission just means that the bacteria replicates its DNA inside, and then it just splits into half and then forms two bacterial cells. And this split, this indent, is called the cleavage furrow, but you don't have to worry about this right now. So they reproduce by binary fission, and because they just split out of one bacteria, they are all identical. Because there is no change in the DNA sequence. They're identical unless, this is the only exception, unless mutation occurs. If while replicating DNA, if there is a mutation, then and only then can they be unidentical, or like very minutely unidentical. So this is how they reproduce. So you might ask that if all bacteria are identical, then how does that make so many bacterial species? Well, not only that, but so if this one should think that if all bacteria are identical, then we can just make one medicine to kill a specific type of bacteria, and then all bacteria can die from it, right? No, because when mutation occurs, the parent cell gives the mutation to its generations. So generations. And because of mutations, this keeps changing as mutations occur generation by generation. This is called the transfer of DNA information, or DNA, from parents to cell, from parents to progeny, is called vertical transfer, because it goes from top to bottom. There is another kind of transfer, which is between two bacterial cells in the same generation. So they can be living together at the same time, and one bacteria can transfer its mutation or its DNA to another bacteria, and this causes bacteria to change and not be identical, and this is called horizontal transfer. So just make sure that you remember the difference between a horizontal transfer and a vertical transfer. The last point is that how do bacteria know how to communicate to other bacteria? And for a long time, we thought that bacteria can't communicate with other bacteria, but in the recent history, we have discovered that bacteria can actually communicate to other bacteria through what is called quorum sensing. Where one bacteria gives signals to other bacteria, and the other bacteria receive that signal. So that is what quorum sensing is. That is how bacteria communicate. And people are researching about if we can stop the communication, then we can stop one bacteria from passing the information or the DNA or the resistant DNA, which is the resistant genes or the drug resistant mutations to other bacteria, so that so that every bacteria doesn't become drug resistant. So that's what they're trying to research on right now. So they communicate through quorum sensing. Communication. So this is all for the microbial metabolism of bacteria. So I hope that you learned something new, and that your doubts were cleared, and thank you for watching.